Good day. The title of my talk is The Effects of Climate Change and Urbanization on the Virulence and Antibiotic Resistance of Vibrio and Cyanobacteria Affecting Seafood Safety and Contact Recreation in the Coastal Zone. My name is Dr. Geoff Scott, along with my colleagues at the Center for Oceans and Human Health and Climate Change Interactions at the University of South Carolina, are glad to make this presentation. We are one of 4 national NIEHS and NSF funded centers on oceans and human health in the United States, and are glad to be discussing this topic with you today in this presentation. Today, I want to talk to you about the interactions of Vibrio bacteria and cyanobacteria that can cause harmful algal blooms in fresh water. If we take a look globally, we are seeing increases in harmful algal bloom due to eutrophication, increased levels of nitrogen and phosphorus, along with climate change that may result in increased temperatures, altered salinities, and pH. And particularly, we want to focus today about freshwater cyanobacteria blooms in estuarine waters. In addition, we also want to focus on the co-occurrence of Vibrio bacteria, which often form strong associations with phytoplankton, including cyanobacteria. And so what we want to do today is talk about these Vibrio cyanobacteria interactions and how this may resultingly affect human health. So if we take a look in terms of climate change and Vibrio bacteria, we are seeing globally that the coastal areas suitable for Vibrio growth are expanding throughout the globe in areas of Europe and the United States, including areas such as the southeastern coast here in South Carolina and North Carolina. In addition to climate change effects, eutrophication is occurring as a result of increased population growth. And we see that the majority of coastlines around the United States have very high levels of population. In fact, more than half of the world's population lives within close proximity to the coastal zone. In the United States, in addition to high population growth, we also have over half of our gross domestic product, an indication of both commercial and industrial activity occurring in the coastal zone. And this leads to increased levels of air and water pollution, such as increased discharges from wastewater treatment plants. You'll notice that we have exposed Vibrio vulnificus to sewage effluent from wastewater treatment plants enriched in nitrogen. The slide on the left shows basically differential expression of gene expression, differentiating between controls in blue and different levels of, controls in red and different levels of nitrogen and phosphorus in our wastewater treatment plant effluent. This in turn leads to increased gene expression for genes involved in energy, membrane transport, metabolism, and stress responses and virulence. In particular, we see hundreds of genes are upregulated and downregulated as a result of this differential exposure to nutrients. This particularly results in increased downregulation in genes involved with motility and increased upregulation of genes involved in biofilm formation in Vibrio vulnificus. This is most noteworthy because this signifies a change in the behavior of Vibrio and may very well, the biofilm formation upregulation of genes signifies increased levels of antibiotic resistance and oftentimes virulence. And in addition to our work in terms of looking at gene expression, we've also developed predictive models to predict levels of Vibrio vulnificus throughout watersheds in the United States, and using a neural network model that integrates climate change models with sea level rise models. We have forecast predicted increases in Vibrio occurrence in terms of these spikes that are optimum increases in optimum growth conditions. And you can see with a 0, 1, 2, or 3 level sea level rise increase, with the 3-foot sea level rise, we see a 230% increase in optimum Vibrio growth conditions. This clearly indicates Vibrios are moving upriver away from estuaries into brackish water areas where they're going to encounter cyanobacteria. In addition, with increased extreme weather, these cyanobacteria are often flushed from these upriver areas into estuaries. In many of these upriver areas and into estuarine areas, we see the increased levels of nitrogen associated with eutrophication can lead to increased toxin production within the microsystem. So therefore, people are exposed then to both increased levels of toxin from the microsystem, as well as from altered Vibrios in terms of antibiotic resistance and virulence. Our toxicology research has focused on looking at the effects of microsystem exposure in terms of its association with illnesses in humans. With 1/3 of Americans being obese, there has been a significant increase in non-alcoholic fatty liver disease. The map at the top left indicates the cyano bloom coverage at a county level, with the red level and orange levels indicating some of the highest bloom coverage. Note as you move along the East Coast, Gulf Coast, the Great Lakes, areas with lots of lakes in Minnesota and along the West Coast, the high levels of bloom coverage. The map on the right indicates the co-occurrence of high levels of bloom coverage in red with high levels of non-alcoholic fatty liver disease. For every 1% increase in bloom coverage, we see a 0.3% increase in non-alcoholic fatty liver disease. Using this information and looking at people that may have the most significant sensitivity to microcystin and Vibrios, we have developed mammalian exposure models that utilize an approach to induce obesity in the mice. Compare that to mice with a lean diet with and without microcystin exposure. The combination of microcystin along with an obese diet basically leads to increased liver injury, along with damage to the kidney, increased inflammation as a result, and alterations in the gut microbiome, which in turn leads to enhanced inflammatory responses that can affect the brain. In addition, we have shown that microcystin can be an endocrine disrupting chemical. We have published a number of studies that have clearly documented these effects. And one of the things we know, this alteration in the gut microbiome and resulting increased inflammation affects the blood-brain barrier. In addition, we have looked at, in weanling mice, we have exposed mice early in life to see if early exposure in life may predispose mice to the development of diabetes and NAFL. And in fact, we see that is exactly the results we see, as we see an increase in insulin resistance and the earlier onset of NAFL. We have also finished experiments now looking at microcystin exposure followed by Vibrio challenge. And we clearly see that when we do Vibrio challenge with our controls microcystin, we do not see major alterations. But what we really see most prominently is when we follow microcystin exposure with Vibrio challenge, we get significant increases in systemic inflammation and in what we call cytokine storm, symptoms very common in sepsis following Vibrio challenge. What this tells us is that Vibrio exposure following Microsystem exposure will be more pronounced and may lead to greater effects from the Vibrio exposure. In addition, as I said, we have shown that Microsystem is an endocrine disruptor. It affects particularly the gonadotropin-dependent follicle maturation and other aspects of female reproductive toxicity. We have also developed an ovary on a chip which uses human cells for rapid screening of samples to look at the effects on the human ovary directly, in addition to our mammalian studies in mice that have documented these effects. Our final aspect of our center is research is focusing on the association of microplastics on Vibrio and Cyanobacteria. Vibrios are often found to be the most significant microbe growing on microplastics. And with microplastics being found in every part of the world, in fact, I think for every one algal cell today, we have 6 microplastic particles to give you the extent and magnitude of microplastic exposure. We have conducted studies which have clearly shown that tire oxidation products and textile fibers are the dominant microplastic found in molluscan shellfish, such as oysters. We have established a reference dose for microplastics for individual oysters, as well as a dozen oysters, which is called a serving, and have made those documented exposures. We also find in studies in the lab with the different microplastic particles that the tire wear particles have the highest bioaccumulation factor. For example, 15 versus a bioaccumulation factor of 1 for the textile fibers. In addition, we have developed depuration techniques to potentially suggest as a mitigation strategy to the shellfish industry for reducing these exposures. And you will note that by 96 hours, we have anywhere from 65 to 80% depuration of the particles. However, the tire wear particles are the slowest to depurate. So in conclusion, we have shown that exposure to Microsystem increases inflammatory disease that affects the ovary, the liver, kidney, gut microbiome, and blood-brain barrier. And in combination with Vibrio challenge, leads to increased systemic inflammation and cytokine storm, very commonly found in sepsis, thus enhancing the effects of Vibrio infections. Our future research will focus now on human health to examine what happens when we challenge mice with microplastic particles coated with Vibrios or Microsystem toxin or both in terms of their effects on mice to see whether they exacerbate some of these inflammatory responses and human health effects we have already seen. So in closing, let me just say that oceans and human health is a One Health concept whose time has come. And we are clearly establishing the linkages between alterations in ecosystem health and human health from these factors such as harmful algal blooms, Vibrio bacteria, and microplastics. Thank you, and I look forward to talking with you more at the meeting as we discuss these topics in more detail.